Hello everyone, myself Professor S.K. Sonone from Kekewa College of Agriculture, Nasik. Today I am presenting the topic under practical syllabus of my introductory microbiology course from Department of Plant Pathology. Today's presentation is exercise number 5, method of isolation of purification and microbial cultures. So how to isolate and purify? Method of isolation of microbial cultures. Okay, object this exercise to isolate the bacterium from liquid sample. Material required for nutrient agar, petri plates, inoculating needle, spirit lamp and water sample. Isolation. So what is meant by isolation? This is the definition of isolation. Process of separating the microorganism from their natural substrate and growing them on culture medium in pure form. This is the definition of isolation. Then procedure of isolation. Prepared the nutrient agar medium. Pour the petri plates with nutrient agar medium at 45 degrees Celsius temperature. Cool down the petri plates so that the media could be solidified. Raise the lid of medium plate only far enough to permit the inoculating needle into enter. Dip the tip of the inoculating needle in liquid sample or bacterial suspension so that bacterial cells stick up tip of the needle. Insert the inoculating needle in nutrient agar plate and streak out the needle on medium by making zigzag or straight or cross lines gently. The bacterial cell present on the needle could enter in the medium and grow. Incubate the petri plate for 3 to 4 days at 30 degrees Celsius. Observations. Observe the plates having different colonies of bacteria and observe the bacterium under microscope to confirm the morphology of that bacteria. Then isolation of fungus. Object to isolate the fungus from diseased leaf sample. Okay, bacteria isolated from liquid sample, fungus isolated from leaf sample. So material potato dextrose agar, because nutrient agar is common media for growing bacteria and potato dextrose agar is a common media for growing fungus. Petri plates, inoculating needle, spirit lamp, blotter paper, mercury chloride solution and distilled water. Procedure. Take fresh sample of diseased leaf showing mango anthracnose symptoms were brought to the laboratory in paper bags. So we taken mango anthracnose symptoms. Anthracnose means mango anthracnose caused by Colitotrichum gliosporus. Small beads of desired size were cut by taking care that each bead contained half infected and half healthy portion. Such beads were then disinfected with 0.1% mercury chloride solution for 1 minute, followed by 3 washings in distilled sterile water to remove the traces of mercury chloride. These beads were then placed on sterilized blotter for drying. Properly dried bits were transferred aseptically in sterilized petri plates containing sterilized solidified PDM medium. The plates were incubated in BOD incubator at 26 plus minus 1 degree Celsius till the fungal mycelium fully covered the surface of the medium. This fungal growth was transferred to PDS plant and maintained as a stock culture for further study. Then purification. Isolation was done. Isolation of bacteria and isolation of fungus. Then when we are isolate any bacteria or any fungus, sometimes there is contamination, means desired organism growing along with the undesired or microorganism. It is called as contamination. So in that cases we have to purify the microbial culture. Then methods of purification. To grow the microbial culture in pure form, only one desired organism grow on the petri plate. It is called as purification. Principle: the microorganisms are required to be grown in pure form, developed from single cell on culture media for their detailed study of morphology, physiology and cultural characters. This is the main principle of purification. So some definitions, some important definitions from this exercise. So first one, culture. What is mean by culture? It is the growth of microorganism on culture medium. Growth of fungus or bacteria, it is called culture. Then colony, growth of microorganism, usually round in appearance on a solid medium resulting from single cell or group of similar cells. Okay, any bacteria grow that time round shape, blur or whitish, whatever it may be, round shape appearance of that bacterial growth, it is called as colony. Then contamination. I already told you, growth of desired microorganisms along with the other undesired microorganism on culture medium. It is called as contamination. These four are culture, colony, contamination and isolation. We already see in the beginning of the definition of isolation. Okay, four important definition in this experiment. Then procedure. During the course of handling, cultures may get contaminated with undesirable microorganisms. Such cultures can be purified by following three methods. First, dilution and plating method, streaking method and spread plate method. First method, dilution and plating method. Dilution means dilution of 1 to 7 water blanks and then dilute your culture and isolate. Okay, we see step by step procedure. Prepared 5 to 7 water blank by pouring 9 ml sterile water in sterile test tube. Prepared nutrient agar medium. Lift full quantity of contaminated bacterial culture with inoculating needle under aseptic condition using laminar airflow cabinet. You know already in the first exercise presentation we see what is the use of laminar airflow cabinet and how they is working this instrument. Dispense the culture in first water blank. This will be 10 raised to 1 dilution. Take 1 ml of suspension from first water blank and pour into second water blank and this will be 10 raised to 1 dilution. Likewise prepared 5 to 10 raised to 7 serially dilutions. Take 1 ml of from 10 raised to 5 to 10 raised to 7 dilution in each pour in petri plates. Pour 15 ml of nutrient agar in the plates containing bacterial suspension. Incubate the plates at 30 degrees Celsius for 3 to 4 days. Then streaking method. Prepared nutrient agar plates. Lift full bacterial contaminated culture. Dispense in 1 ml of sterile water in test tube under aseptic conditions. Using laminar airflow cabinet, dip the inoculating needle into the bacterial suspension and streak on the agar medium by making zigzag, straight or cross lines. Incubate the plates 30 degrees Celsius at 3 to 4 days and finally observed. Select well isolated colony and transferred on nutrient agar medium for purification. Then third method, spread plate method. Prepared nutrient agar petri plate. Prepared suspension of contaminated bacterial culture in sterile water. Dip L-shaped bent glass rod in a suspension and spread on the agar plates. Incubate the plates at 30 degrees Celsius and for 3 to 4 days. Observe selected well isolated colony and transfer on the nutrient. Purification of fungal culture. Okay, these three methods we've studied. That is purification of bacterial culture. Now purification of fungal culture. Following two methods. First method, single hyphal tip method. In this method, initially growing hyphal tip is selected from bacterial contamination and transfer in the slant to obtain pure culture. Grow the colony of fungus on the PDA medium. Select the single growing hyphal tip of the mycelia. With the help of microscope and mark it with the glass marking pencil. Cut the mark portion with the help of cork borer and transfer on into the PDA slant medium and incubate at 27 degrees Celsius and observe the pure growth of fungal culture. Then single spore isolation method. How to isolate single spore of fungus. Prepare the spore suspension from the sporulating fungus. Then add about 10 ml of spore suspension in 250 ml of liquefied potato dextrose agar medium when the temperature of medium is around 40 degrees Celsius. Add the medium in sterile petri plate, allow to solidify, make a slant. After the solidification of the medium, locate the single spore with the help of microscope and mark with the glass marking pencil. Cut the mark portion with the help of cork borer and transfer it on the PDA slant. Incubate the slant at 27 degrees Celsius for a week for observe the pure growth of culture. So thanks for listening this presentation. Practical exercise is very important. Isolation and purification. Thank you very much.